Imagine a petri dish. Inside, you see microscopic particles, a chaotic soup of biological debris. Then something enters the frame. It's a tiny, blob-like cluster of cells, scuttling with purpose. It has no brain, no nerves, no muscles as we know them. And yet, it moves. It finds the tiny particles and begins to push them, gathering them into neat, tidy piles. It works. You watch as a scientist cuts this tiny creature nearly in half. You expect it to die. Instead, the two halves pull themselves back together, stitching themselves up until the creature is whole again. It heals, and then it goes back to work. What in the world are you looking at? It is not an animal you would find in a pond. It is not a machine you would find in a factory. It is 100% living cells, taken from a frog embryo. But it is also 100% a robot, designed by an artificial intelligence. This is a xenobot, and it is the first true living machine organism. It is a creature that should not exist, and it is a perfect messenger from a future that has already arrived. For all of human history, we have lived in a world of clean categories. There was the natural, the world of trees, animals, and our own bodies. This was the world that grew. And then there was the artificial, the world of hammers, cars, and computers. This was the world that was built. One was the product of blind evolution, the other the product of conscious design. That line, that fundamental boundary that separated life from non-life, the grown from the built, has just been erased. It's important to understand what this isn't. This is not biomimicry. For centuries, we have copied nature. We saw a bird's wing and, understanding aerodynamics, we built an airplane. We admired the strength of spider silk and we synthesized new polymers, like nylon, that mimicked its properties. We were learning from nature's design, but we were still building our machines out of metal, plastic, and silicon. The xenobot is not a copy. We didn't build a robot that looks like a cell. We programmed the cells themselves to be the robot. This is also not a cyborg in the traditional sense. When we think of a cyborg, we imagine a human with a robotic arm or a pacemaker. That is a fusion of a complex organism and a distinct mechanical part. You can still point to the line. You can see where the flesh ends and the metal begins. It is an additive technology. The machine organism is a foundational technology. The line is not just blurred. It's gone. The machine and the organism are one and the same. The engineering is not an attachment. It is written into the living matter itself. The xenobot is an entirely new category of being. Its software is the 4-billion-year-old operating system of DNA. Its hardware is the soft, living, self-healing chassis of a cell. But its purpose, its form, that came from us. Or more specifically, it came from an AI that we instructed. We have liberated living matter from the evolutionary destiny it had for eons, to become a frog, and given it a new one, to be a tiny, living machine. This breakthrough forces us to ask a profound and unsettling question. We have spent centuries building machines out of inert matter, telling them exactly what to do. We grew comfortable with that power. But what happens now? What happens when our technology is no longer made of steel and silicon, but of the same self-replicating, self-healing, adaptive matter as us? What happens when our technology isn't just built, but grown? And what happens when that living technology starts to have ideas of its own? Imagine a computer screen covered in lines of code. The letters $G$, $C$, $A$, and $T$. You're looking at a text file, a simple string of data. Now, imagine you could hit compile, and that code would fold itself out of the screen, spring to life, and start eating sugar to build a microscopic engine. This isn't science fiction. This is the reality of synthetic biology. For decades, we thought of life as a magical, unknowable essence. Now, we finally understand it for what it is, the most sophisticated information processing system in the universe. And the machine part of our machine organism is born from one simple fact. We can now write and edit this code. We have become biological engineers. The machine is not metal or plastic. It is our intent, impressed upon a living system. And we do this using a toolkit of breathtaking power. The first tool is synthetic biology. You can think of this as genetic programming. This is where we write new code, new DNA from scratch. Scientists no longer have to just find a useful gene in nature. They can design one on a computer. They can write a genetic program that instructs a cell to perform a specific task. The most famous example of this came in 2010. Scientists led by Craig Venter built an entire bacterial genome, all 1.08 million base pairs, from bottles of chemicals. They typed out the genetic code of a bacterium, synthesized that DNA, and then booted up a dead cell with this new artificial software. The cell came to life. It began to feed, to grow, to replicate, all under the control of a genome that was written by humans. This is the power of read-write biology. We can now design a microbe like yeast or coli dollar to be a living, single-celled factory. We give it a new string of DNA code. This code instructs the cell to eat sugar, and instead of producing just alcohol, it produces, well, almost anything we want. We have bacteria that excrete high-performance biofuels. We have yeast that brews the proteins for spider silk, one of the strongest materials known to man. We have microbes that produce the pigment that makes a plant-based veggie burger bleed, like real meat. These are organisms, yes. They are alive. But they are also machines designed and programmed for a human purpose. The second tool is CRISPR-Cas9. If synthetic biology is like writing a new Word document from scratch, CRISPR is the find and replace function. This is genetic editing. For years, editing DNA was like trying to edit a single word in a 3 billion-letter encyclopedia, with the book chained shut in the dark. CRISPR changed everything. It's a molecular machine, a set of scissors that we can program. We give it a search query, a small piece of guide RNA. It scans the entire vast library of an organism's DNA. When it finds the exact sequence it's looking for, it cuts. Where did this miraculous tool come from? We didn't invent it. We found it. CRISPR is an ancient immune system used by bacteria to fight off viruses. They remember a virus by snipping out a piece of its DNA and storing it in their own genome. If that virus ever attacks again, the bacteria sends out the CRISPR-Cas9 protein with that memory file, finds the invader's DNA, and destroys it. We hijacked this billion-year-old defense mechanism and turned it into the most precise editing tool in history. We can now go into a living cell and change a single letter of its code. We can fix the mutation that causes sickle cell anemia. We can make a crop resistant to drought. Or we can edit a mosquito so it's physically incapable of carrying the malaria parasite. It is the tool that allows us to edit the organism part of our hybrid. The third tool is tissue engineering and bioprinting. This is the 3D printer for life. A normal 3D printer builds an object layer by layer out of hot plastic. A bioprinter works the same way, but it uses a miraculous substance called bioink. This bioink isn't ink at all. It's a special, nutrient-rich hydrogel that is packed with thousands of living human cells. A scientist designs a 3D model of a structure on a computer, a human ear, a patch of skin, a small section of a blood vessel. The bioprinter hums to life, and its nozzle extrudes the bioink, not in a random blob, but in a precise, layered scaffold. Prints a layer of cells, then a layer of supporting gel, then another layer of cells. At first, it's just a delicate, wet scaffold. But then the magic happens. The cells are alive. They don't just sit there. They talk to each other using chemical signals. They begin to organize. They fuse. They specialize. The stem cells that were printed next to each other differentiate, becoming a functional tissue. Scientists have printed patches of heart tissue that, after a few days in a bioreactor, begin to beat. On their own. A rhythmic, living pulse. This is the bridge. This is where the designed scaffold from the machine meets the autonomous function of the organism. We design the architecture, but life builds the home. We have the tools. We are no longer just observers of biology. We are its architects. We can write new life, edit existing life, and print living, functional structures. We have the machine part of the equation mastered. Now, let's see what happens when we apply these tools to create the organism hybrids that are already changing our world. Imagine holding a tiny, breathing lung in the palm of your hand. It's not a real lung, but it's not a fake one either. It's a clear, plastic chip, the size of a USB drive, and inside its tiny, intricate channels, living human lung cells are growing. A vacuum pump gently stretches and relaxes the tissue, making it breathe. A liquid blood flows past it. You can puff cigarette smoke over this chip and watch, in real time, as the cells become inflamed. You are not looking at a computer simulation. You are looking at a true machine-organism hybrid. This is an organ on a chip, and it is a revolution. For decades, testing a new drug was a slow, expensive, and often cruel process. First, we test it in a flat, plastic petri dish. But cells in a flat dish don't behave like cells in a 3D dynamic human body. So, if the drug passes that test, we move to animals like mice or primates. But a drug that is safe for a mouse can be deadly for a human. Many promising drugs fail in human trials after billions of dollars have been spent. The organ on a chip solves this. It is the perfect hybrid. The machine is the microfluidic chip, designed by engineers with incredible precision. It has channels for air, channels for blood, and mechanical systems to mimic the body's physical forces. The organism is the living tissue grown from human stem cells that lines these channels. We now have lungs on chips, livers on chips, guts on chips, and even hearts on chips. We can test a new drug on a living, breathing, metabolizing human system without ever endangering a human life. We can even link them together, a human on a chip, to see how a drug affects the liver, is processed, and then affects the heart. This isn't just a better model. It's a new way of doing science, a fusion of silicon and cell. Now let's go back to the creature that started our journey, the Xenobot. We hooked you with its image. Now, let's dive into the profound story of its creation. It was born from a collaboration that is itself a hybrid. You had biologists at Tufts University, masters of the organism, and computer scientists at the University of Vermont, masters of the machine. The biologists knew how to harvest and grow two types of cells from a frog embryo: passive skin cells and active heart muscle cells. The heart cells are natural motors. They contract rhythmically, all on their own. The computer scientists were given a challenge. An AI was given a physics simulator with these two cell types as its building blocks. The AI's task was simple: design a new organism that can move on its own. The AI had no preconceptions. It didn't know what an animal looked like. It just ran thousands and thousands of evolutionary simulations, tried random shapes, put heart cells here, skin cells there. Most failed. But the fittest designs, the ones that moved even a little, were bred together. After weeks of simulation, the AI produced blueprints for life forms that looked nothing like anything on Earth. They were bizarre, alien, and, according to the simulation, functional. Now, the biologists took over. They used microtweezers to sculpt the living frog cells, following the AI's blueprint. They put the heart cells exactly where the AI told them to, creating a chassis of skin cells with a motor of heart cells. And the second they were finished, the things woke up. They began to move, just as the AI had predicted. This is a creature with a 100% biological genome, but a 100% artificial body plan. It is a living machine, a biological robot, born from the mind of an artificial intelligence. But the story doesn't end there. There is a third kind of hybrid. Not just biological parts in a machine structure like the organ on a chip. Not just biological parts designed like a machine like the Xenobot. This third type is the most intimate fusion of all: machine parts inside a biological structure. This is the new field of cyborg tissue. The problem with merging electronics and biology is fundamental. Electronics are rigid, dry, and fixed. Tissues are soft, wet, and constantly growing. How do you merge the two? The answer is you don't. You build them together at the same time from the ground up. Scientists are now experimenting with 3D bioprinting, but they are adding a new ink to the printer. Alongside the bioink of living cells, they are printing flexible, nanoscale electronic sensors and wires. Imagine a piece of lab-grown heart tissue. But as it grows, It incorporates a soft, web-like electronic mesh into its very fabric. This mesh is not a pacemaker attached to the heart. It is part of the heart. It's a cyborg tissue. This integrated electronic nervous system can monitor the health of the tissue from the inside, cell by cell. It can feel the beginnings of an abnormal heartbeat, an arrhythmia, long before any external sensor could. And it can deliver a tiny, precise electrical pulse to the exact cells that are misfiring, correcting the rhythm before it becomes a problem. The hybrids are no longer theoretical. They are here. We are designing new life forms in a supercomputer. We are building miniature, functional human organs that live on silicon chips. We are weaving electronic sensors into the very fabric of our living, beating tissues. We have become the masters of design. But this mastery is about to collide with a force far older, far larger, and far more powerful than any design we can imagine. We are about to meet the ghost in our own machine, evolution. For 4 billion years, one process has shaped every living thing on this planet, from the bacteria in your gut to the brain reading these words. This process is blind. It is slow. It has no goal, no intelligence, no forethought. It simply filters, tinkers, culls. The weak, the slow, the unfit are removed. The lucky, the strong, the well-adapted survive and pass on their traits. It is natural selection. It is the raw, untamed engine of all life. And we, in our brilliance, have just created designed life. We have built our perfect, precise, predictable machine organisms. What happens when our new, clean design meets the ancient, chaotic master? This is the central, terrifying, and exhilarating tension of our new age. A machine is designed. An engineer builds a car engine. They know where every bolt, every piston, and every wire goes. It operates according to fixed, top-down rules. It does what it is told and only what it is told. An organism, on the other hand, evolves. It is a bottom-up system. A bird is not designed for flight. It is the result of millions of generations of random mutations and non-random selective pressures. It adapts. It changes. It survives in a world its ancestors could not. Our new machine organisms are both. We designed the Xenobot, but it is made of living cells. It is a biological entity. And all biological entities, whether we like it or not, are subject to the fundamental rules of biology. They are subject to evolution. At first, we thought we could control this. In fact, we even turned evolution into a tool. This is a Nobel Prize-winning field called directed evolution. It is quite literally speedrunning natural selection in a test tube. Imagine you want to create a new enzyme, a protein that can eat plastic, a task nature never had a reason to invent. You take the gene for a similar enzyme. You put it in a beaker and make billions of copies. But you use a sloppy copying process, one that introduces thousands of random errors or mutations. You now have a library of billions of slightly different mutant enzymes. You then test all of them. You pour them onto a piece of plastic. Which ones eat it the fastest? You find the top 1%, the fittest survivors. You take those and you repeat the process. You copy the winners, sloppily. You test them again. You find the new winners. You do this over and over. In a single afternoon, you can force a protein to undergo what might have taken nature 1,000,000 years to achieve. You have used the power of mutation and selection as an engineer's tool. You have directed evolution. We are using evolution to build our machines. But here is the twist. What happens when evolution surprises the designer? Let's return to the Xenobot. The scientists who designed them had a specific purpose in mind. Move push particles. They designed them. They built them. They were done. But then, they got curious. They put a swarm of these AI-designed living robots into a new petri dish, one that was filled with thousands of loose frog stem cells. They just wanted to see what would happen. What they saw next was one of the most profound and unsettling discoveries of the 21st century. The Xenobots, which were only programmed to move, started working together. They began to move in circles like tiny shepherds. They corralled the loose cells, pushing them into little piles. They gathered them, compacted them, and then they left. The scientists watched, stunned, as these new, hand-built piles of cells fused. They grew, they organized, and within a few days they became new, functional, swimming xenobots. Let's be absolutely clear about what this means. The xenobots found a way to replicate. It wasn't biological replication like a frog laying an egg. It was something new, kinematic replication. It was a robot building another robot, out of living parts. The scientists did not design this. They did not program this. It was an emergent behavior. The living part of the machine, faced with a new environment, found a way. Life, as it is so often said, finds a way. The machine had a design, but the organism had an imperative. And the organism won. This leads us to the final, sobering reality. This is environmental selection. This is what happens when we release one of our creations. Imagine we take our perfectly designed bacterium, the one we built with directed evolution to eat plastic. It works perfectly in the lab. It is our perfect living machine. So we release it into the Great Pacific Garbage Patch. The second we do that, we lose control. The lab is a clean, simple, controlled environment. The ocean is not. The ocean is a chaotic war zone. It is filled with other bacteria competing for the same food. It is filled with viruses that have evolved for eons to attack bacteria. It has changing temperatures, changing salinity, and sunlight. Our perfect, designed machine is immediately and violently subjected to natural selection. It must adapt or die, and it will adapt. Random mutations will occur as they do in all life. The bacteria that, by sheer chance, has a mutation that makes it better at competing for food or better at resisting a virus will survive. The ones that don't will die. The organism we get a year later will not be the organism we released. It will be its evolved descendant. Its designed code will have been edited and overwritten by evolved code. Maybe its properties will change. Maybe it stops eating plastic. Maybe it develops a new appetite for something else. The machine we built has been sacrificed to the evolution we cannot control. It has become a new, wild branch on the tree of life. And we are its creators. We have opened Pandora's box. We have merged the clean, top-down blueprint of the designer with the messy, bottom-up, unpredictable power of evolution. This is not a distant, theoretical future. This feedback loop where our creations adapt to the world, and we, in turn, are forced to adapt to their adaptations is already here. It is happening in our most advanced hospitals, saving lives. It is being developed to clean our worst environmental disasters. And it is being built into the very foundations of our cities. Let's start with medicine. Your body is already a war zone of evolution. When you get a bacterial infection, you take an antibiotic, a designed chemical weapon. But in a population of billions of bacteria, a few, by random chance, will have a mutation that makes them resistant. They survive. They replicate. Soon you have a superbug that the drug can't touch. We are in an evolutionary arms race with microbes. Now consider cancer. Cancer is a terrifyingly personal evolutionary battle. It is your own cells mutated. They are evolving, adapting, and becoming resistant to our designed chemical drugs, chemotherapy. For decades, we have been fighting an evolving enemy with a static weapon. And far too often we lose. The new strategy? We must fight evolution with evolution. This is CAR T-cell therapy. It is a living drug. It is perhaps the most profound machine-organism hybrid in existence today. Doctors take a sample of your blood. They filter out your T-cells, the soldiers of your immune system. In the lab, they engineer these cells. This is the machine part. Using a disarmed virus, they insert a new artificial gene into your T-cell's DNA. This gene is a blueprint for a new protein, a chimeric antigen receptor, or CAR. This CAR is a synthetic targeting system, designed perfectly to find and lock onto a specific protein that only exists on the surface of your specific cancer cells. They grow billions of these new cyborg T-cells in the lab and then infuse them back into your body. They are now your cells, but they are also a designed living medicine. They are machine organisms with a single mission: hunt and kill. They swarm your body, find the cancer cells, lock on, and destroy them. The results have been miraculous, curing patients with incurable blood cancers. But here is the evolutionary feedback loop. These CAR T cells don't just work. They live. They multiply inside you. They form a living, evolving memory in your immune system. If years later a few of those cancer cells try to come back, this living medicine is still there, patrolling. It adapts with you. We have created a personal, evolving immune system to fight a personal, evolving disease. Now let's look at the environment. Our dumb, 20th century machines left a legacy of pollution. Oceans choked with plastic, skies filled with C02, and landscapes poisoned by oil spills. We are now trying to build living machines to clean up our mess. We've discussed the bacteria designed to eat plastic or oil. This research is happening in labs all over the world. But this is where the feedback loop becomes a problem. This is where the terror of Act 4, losing control, becomes a real-world engineering challenge. If you release a plastic-eating microbe into the ocean, how do you know it will only eat plastic? How do you know it won't evolve to outcompete the natural, vital microbes at the base of the ocean's food web? How do you stop it? This has created an entirely new field of research: genetic firewalls and kill switches. We are now in an arms race with our own creations. For every machine organism we design to do a job, we must also design a control mechanism. For example, scientists are engineering bacteria that must have a specific artificial amino acid to survive, a food that doesn't exist in nature. We can spray this artificial food over an oil spill along with the bacteria. The bacteria thrive, they eat the oil. When the oil is gone, we stop supplying the artificial food. The entire population, unable to live without its designed weakness, starves and dies. We must anticipate how our creation might evolve to bypass that control. What if it mutates and learns to synthesize that amino acid itself? We must design a second kill switch. And a third. We are now designing machines that are programmed to die, to counteract the organism's imperative to live. Finally, let's look at materials. Our world is built of dead, inert things: concrete, steel, plastic. When they break, they stay broken. Entropy always wins. But what if our materials were alive? What if our buildings could heal themselves? This is not a what if. This is self-healing concrete, and it's already being used. When engineers mix this special concrete, they add 2 extra ingredients: tiny, microscopic capsules containing bacterial spores, like a dormant seed, and a packet of their food, calcium lactate. The concrete is poured. It hardens. The spores lie dormant, inert, for decades. They are, for all intents and purposes, just another part of the machine. Then, one day, a microcrack forms in the foundation. Water seeps in. This water is the trigger. It wakes up the dormant bacteria. The spores germinate. The bacteria find their food packet and begin to eat. And their metabolic byproduct, their waste, is limestone, calcite. The bacteria secrete limestone into the crack. They fill the damage they are living in. They heal the concrete from the inside out, and then, their food source gone, they go dormant again. This is the perfect, beautiful, simple machine organism. A dumb machine concrete, infused with a living organism bacteria, that performs a single, designed function: healing. The feedback loop is complete. We build the machine. The organism maintains it. Look at your own hand. It is a masterpiece. It is the product of 4 billion years of blind, stumbling, chaotic, and beautiful evolution. It is a thing that grew. Now look at the screen you are watching this on. 2 is a masterpiece. It is the product of a century of intelligent, purposeful, and brilliant design. It is a thing that was built. For all of human history, these 2 empires, the grown and the built, the natural and the artificial, existed in separate universes. We were subjects of the first and masters of the second. Until now, we are the generation that is witnessing the end of that separation. We are the architects of the bridge between them. And as that wall comes down, we must accept that nothing will ever be the same again. We began this journey in a petri dish, watching a microscopic puzzle. A living robot, the Xenobot, a creature designed by an AI and built from living frog cells. A creature that should not exist. We learned that this creature was not a miracle, but the product of a new and awesome technological power. We have become genetic programmers, writing new life from scratch with synthetic biology. We have become genetic editors, rewriting the code of any organism at will with CRISPR. We have become biological architects, 3D printing living, functional tissues and organs. We saw the hybrids that are already here. The organ on a chip, a miniature, breathing human lung living on a silicon chip. The cyborg tissue, a beating heart with an electronic nervous system woven into its very fabric. And then we faced the twist. We learned that life is not a quiet medium. It does not just obey. It adapts. We watched in awe as our Xenobot creations invented a new form of replication, a behavior their designers never intended. We confronted the reality that when we release our living machines into the wild, we are sacrificing our design to the ancient, untamable power of evolution. We saw this new reality, this feedback loop already at work. We are creating living medicines, like CAR-T cells, that evolve inside our own bodies to hunt our most evasive diseases. We are designing living solutions, bacteria to clean our world, and then designing kill switches to control them, locked in an evolutionary arms race with our own creations. We are building living concrete that heals itself. The old question, is it natural or is it artificial, is now scientifically and philosophically obsolete. It is a meaningless distinction. A CAR-T cell is 100% your natural cell. It is also a 100% designed and artificial piece of medical technology. It is both. A Xenobot is 100% frog DNA. It is also 100% an AI-designed robot. It is both. The line is gone. We have entered the age of hybrid life. We are not just using nature or imitating nature. We are fusing with it. We are becoming its co-authors. For the entirety of its history, evolution was a force acting upon humanity. It was the sculptor, we were the stone. It was the slow, blind, powerful force that shaped our world and our very bodies, and we were powerless to stop it. Now we have picked up the chisel. We have harnessed evolution in our test tubes. We have directed it. We have sped it up 1,000,000-fold. We have made it a tool of our own design. And in return, our designs, these new living, breathing, healing, working machine organisms, have been thrown into that ancient, chaotic river of evolution themselves. We have created a new feedback loop that will define the next 1,000 years of human history. The machine and the organism are one. The blueprint of design and the engine of evolution are now locked together, accelerating into a future that we can design, but can no longer fully control. We have rewritten the definition of life. And in doing so, we have just started to rewrite the definition of ourselves.